presenting. There we go, we're recording. And yeah, we've got some cool research. If you're a long-time Asheville Science Tavern attendee, you may recognize some of the names here and some of the topics. We've been graced with pitcher plant talks before, and we're excited to hear where this research has gone now. All right, so I'll give it over to Becca Hale. Or I guess Jen, whoever student this is.
This one's Jen's.
Good evening,
Okay.
everyone. I'm Jen Rhode Ward. I'm a professor of biology at the University of North Carolina, Asheville, and I've had the pleasure of working with our first speaker, Claudia Prieto Alcaide, for more than a year on a project on pitcher plant conservation. And she has been working on the population genetics behind two species of conservation interest and their hybrids. And so let me tell you just a little bit about Claudia. So Claudia is originally from Spain, from Madrid, and she came to North Carolina specifically to attend UNC Asheville on a track scholarship. So she is an amazing long jumper. If you Google her name, you'll see pictures of that. So she's been working on this project for more than a year. She's going to graduate next year, and she just received our department's Excellence for Undergraduate Research in this past year. And when she finishes with us at UNC Asheville, she will be going on to graduate school to study molecular biology and genetics, building on some of the skills that she's built up for herself during this project. So I'm really pleased to introduce you and to let you hear about her last year plus of research.
Thank you for the introduction. But yeah, I'm very excited to show you what I've been working on for the past year, which is confirming hybridization status between two North American pitcher plants, Cersei johnsiae and Cersei Puerpria varmontana. And let's see. So a little bit about what hybridization is. Hybridization occurs when two parental species produce offspring, which would be first generation hybrids or F1 hybrids. And then these hybrids can breed with each other, creating second generation hybrids or F2 hybrids, or they can go back to breeding with their initial parental species in a process known as backcrossing. But as most things, this can have both positive outcomes and negative outcomes. And the positive outcomes mainly include speciation and adaptive genetic diversity. And the negative outcomes include integration with non-native species, which is an example that we already see in native bittersweet and Asian bittersweet in this country. And then there's also loss of genetic identity in species that might already be endangered. So these are the two pitcher plants that I've been working with. Cersei johnsiae is also known as Jones pitcher plant. It is only found in 10 small populations. 4 of those populations are in North Carolina and 6 are in South Carolina. But there used to be over 16 populations, and they've been extirpated between now and like 1988. So this is why this species is considered to be federally endangered by the Fish and Wildlife Service. And my other plant is Serosonia Puerpria varmontana, also known as a mountain purple pitcher plant. It is found in around 20 populations in North Carolina, South Carolina, and Georgia. And it is currently a federal species of concern. But if we look at these maps by nature reserve, we can see that internationally they've been labeled as imperiled and critically imperiled. So it is a plant that internationally, both of them are pretty endangered and found in very few places in the world. So the threats that they both overcome are overcollection, poaching for the rare plant trade, and habitat loss, because the habitats where these plants grow are mainly being used for agriculture and development. Sorry. So why is it important to study hybridization between Serosonia johnsii and Serosonia purpurea? These two species coexist. So in one of the sites that we studied, they've been growing together in the same populations since before 2005 and potentially since 1960. But in the other populations, they initially did not grow together, but in order to try to preserve Serosonia johnsii, the Fish and Wildlife Service has taken some of this Serosonia johnsii samples and outplanted them intentionally into Serosonia purpurea populations. So this is how they have possibly started to hybridize. And there have been phenotypic hybrids observed, but they have not been genetically confirmed until this research. And the problem with this hybridization is that it can lead to competition. So those hybrids might outcompete the parental species. And hybrids also don't have any official status. So when two plants that are federally listed hybridize, they might not have any official endangered status. So my main research questions are how much genetic diversity is found in these three populations that we studied, and if the phenotypic hybrids are actually genetic hybrids. In order to test this, we first began DNA extraction and quantification. DNA was extracted with a modified CTAB method, and then it was quantified using a nanodrop. And on this table over here, we can see that the number of samples from each species in each population that we analyzed. Then in order to amplify microsatellite loci that were specific to Serosonia johnsii or Serosonia purpurea or both, we performed PCR. And then these PCR products were run on 3% agarose gels. And this was used to confirm if the genetic marker had amplified. And sometimes this was the last step for some of the microsatellite markers we use. But in other cases, we had to go into fragment analysis and data analysis, which I'll explain in a second. I wanted to kind of give you a really quick background on what microsatellite loci are. They are repeating DNA segments. And in my case, we're looking for dinucleotide repeats, which mean they would be 2 base pairs long. And they are typically found in the DNA non-coding regions. So these are the 7 microsatellite loci that I used to obtain my results and for my research questions. Serosonia or loci 10 through loci 45 were used to determine the hybrid index between these two pitcher plants. And then 10, 5, and 50 were used to determine population level genetic diversity. And yeah, then we went on to fragment analysis. So samples were multiplexed and then they were sent out to NC State for fragment analysis. And then we used Genius to bin those fragments and estimate the size of those microsatellite repeats. And to determine the data that we got from Genius, we used 2 R packages. The HYST package was used to determine hybrid index, and the polysat package was used to analyze population level genetic diversity. So these are the first results that I got. And they show that allelic diversity varies by species and location. So I have this data for 3 loci, but I'm presenting data from just one of the genetic markers because it has the most complete data set, and the other ones have some missing data, so I felt like it could have skewed the results. We can see in the chart that site CM has more diversity for Serosonia purpurea because this is a numerically dominant species here. Site MB has more diversity for Serosonia johnsii because Serosonia johnsii is a numerically dominant species in this site. And then hybrid diversity is highest at site RL, and this is because there are more hybrids in that site than in any of the other sites. But we can see that allele diversity is pretty high for both parental species at each locus. The next results we obtained are a box plot showing the ancestry of the hybrids. The y-axis is showing the ancestry of all the hybrids studied, where 1.0 would mean that the plant is a pure Puerpria, so all the alleles in that hybrid would be, well, they wouldn't be hybrids, all the alleles in that plant would be Puerpria alleles, and then 0 would mean that a plant is a pure Serosonia jonesii. So across all three sites, the median hybridization index is 50-50, which means that there's equal amounts of both parental species in those hybrids. And based on this little table over here, the results would mean that the hybrids are either first or second generation hybrids. Then when we break it by site in this box plots, we can see that these are very interesting results. These are probably the most interesting results that we observed. And site CM Puerpria is the most numerically common species, but the hybrids happen to be more Serosonia jonesii. And MB site, jonesii is most commonly numerically, but the hybrids are more Serosonia Puerpria. And at the RL site, both species are abundant, so the hybrids have like a really broad range of ancestry. And then finally, we created this bubble plot, and it examines the heterozygosity versus ancestry, and this would more accurately tell us the generation that these hybrids belong to. So the size of each bubble indicates the relative abundance of individuals with the same combination of heterozygosity and ancestry. And as we can see where it says F1 and F2, if those hybrids have been first or second generation hybrids, there would be a lot of bubbles around that area. But we see most of the bubbles down at really low heterozygosity levels. So this could mean that there is back crosses, meaning that the hybrids are going back to breed with their initial parental species rather than breeding with other F1 or F2 generation hybrids. And just to wrap up the results a little bit, we see that allele diversity is relatively high overall. So at sites where that taxon is most abundant. And why is this? This could be due to a bigger gene pool and the fact that there are more individuals to sample. Then hybrid ancestry varies, and these are the very interesting results that we got. Less abundant taxon contributes disproportionately to ancestry. There's a lot of reasons why this could happen. It could be to pollinator preference, selection, or possibly to competition with parental species. If at a site there are many more Serosonia Puerperias than Serosonia, the plant might feel like it has less competition by choosing alleles from Serosonia jonesii rather than Serosonia Puerperia. And then hybrids seem to be back crossed. So this low heterozygosity values could be skewed though. And this could be due to the fact that 5 taxonomically informative loci were used, but only 4 were used for presence or absence, and only one of the microsatellite loci was used to determine the heterozygosity value. And the next steps could include a complete analysis of the population genetic data. So just using every microsatellite loci that we had for every sample, which we didn't have time to do in just 1 year. We could observe pollinator visitation patterns, and then we could provide conservation recommendations to the U.S. Fish and Wildlife Service, as they would need to take care they would need to watch what sites they outplant Serosonia jonesii into to make sure these plants are not constantly hybridizing and they outcompete the parental species, and maybe just check the pollinators to make sure that these plants are not hybridizing. And these are my references. And I want to say Thanks to everybody that contributed in this project and made it possible. Thank you to my faculty advisor, Dr. Rhode Ward, my committee members, Professor Kennedy, Dr. Hale, and Dr. Meeks. I couldn't have done this project without the funding from the U.S. Fish and Wildlife Service and the UNC Asheville Undergraduate Research Program, and my friends, Tiana Miller, Grace, Yasmin, who really helped me throughout this year, and they really made sure that I had as much information as I could to present today. So thank you so much.
Great, thank you. Looks like we've got a question in the chat. What is the cause of these species sharing locations? Is human activity involved, and is it expected that similar species would hybridize naturally? So the three-parter.
So the reasons why these species are found in the same populations is it could be due to, in some sites they were already initially coexisting, maybe since like 1960s. So they already grew together, but in some cases, since Serosonia johnsiae was going extinct, the Fish and Wildlife Service tried to outplant Serosonia johnsiae into other sites, and because both Serosonia purpureas and Serosonia johnsiae is like the same type of habitat, all the sites that they tried to outplant Serosonia johnsiae into were already taken over by Serosonia purpurea, so they still outplanted them in there. So that's why they've been growing together. And yeah, as I was doing my research and writing my research paper, I did read a lot about Serosonia species freely hybridizing with each other. So it's not just these two Serosonia species. Most Serosonia species really, like, I guess they have the tendency of hybridizing with other Serosonia species. So it's not just these two plants.
Great, thank you. Seems like you've done your research. I have a quick question for you, and it's more of a, I actually have two. One is a general one, and then one is a little more specific about the statistics. But I'm curious, what are the main pollinators for these pitcher plants? And then my specific question is for the microsatellite analysis, do you need an even ratio of microsatellites that are available for specific species to get the correct hybridization index analysis?
So for the first question, there is not a lot of research done on Serosonia species pollination, but from the few papers that do mention it, they say that bees are the main pollinator. And then the second question, you asked if there has to be an even number of microsatellite loci for each of the species.
Right, so do you need an even ratio of microsatellites to have the correct hybridization analysis?
Yeah, probably around the same specific hybrids for Cersei johnsiae or Cersei Puerpria. I mean, we probably didn't have the exact same number of both, but close to. There were at least there were many more primers than we ended up using for this results. We just didn't have time to test them all, but we do have a few microsatellite loci that are specific to Cersei johnsiae and around the same number that are specific to Cersei Puerpria. But then there are a lot also that should amplify for both. So I think we have a even number of like all three of those satellites.
So I just want to to Harris's question. So for Claudia's work on hybrids, the markers that she used had specific patterns for each of the two species. So for the population level work, I think it would be important, to your point, Harris, to have like equal markers to look at population diversity. So for the population level, there's some markers that only work in one species, like Claudia said, and some only in the other. But for the work that Claudia did on the hybrids, all of those genetic markers would amplify one way for one species and one way for the other.
Cool, great, thank you. Open up the floor, see if anybody else has any questions. All right, well, great. Thank you so much. That was excellent.
Thank you.
I guess now we'll welcome our next speaker for our May doubleheader. Becca, would you introduce
Yeah,
Todd?
I'll introduce him and I'll provide a segue as well. We've been, Jen Ward and our colleague Caroline Kennedy and I have been working on pitcher plants for probably 8 to 9 years now. And we've been interested in this problem of hybridization and how we can help the folks who actually make the conservation decisions and the management decisions, how we can help inform, what they might want to do. And Claudia's was the first study to really take that genetic approach and really help us figure out who is a hybrid and who is not. And so that's been really important, is a really important part of our work, to have that piece of it. And as she described, there's a huge amount more hybridization than we realized, which suggests that they've been hybridizing for a very long time. And as Claudia said, large bees like bombus bees and other bumblebees are thought to be the primary pollinator of probably all the Serosonia, and they are fairly generalist pollinators. And so there was a question in the chat about hybridization happening in nature, and there are anecdotal comments about that in a lot of the literature on these species, because facilitated by their being visited by kind of all the same pollinators. Another aspect of our work that Jen Ward has been spearheading has been to actually try to quantify the consequences of cross-pollination. Pollination, yeah, that sounded weird to me for some reason. And we're not including that in today's work, but maybe she has talked about that before. I'm not sure what the topic of her previous talk was. But our next speaker is Todd Brasseur, who's been working with us for, let's see, this will be his 3rd summer with us. Todd is a UNCA student who is graduating this calendar year. And he picked up on a project that previous student, Wayne Morgan, had worked on. And this was to figure out how can we actually try to tell apart hybrids in the field, because we can't do genetics in the field, at least not in 2022. So we recruited Todd, and Todd has been a student in the biology department since I lost my bio of him, for a few years now, and he moved to Asheville from Michigan, where he had a previous career in sound design and sound engineering, and then shifted gears to study biology with us. And so for the last couple of years, he's been working on this project, and he has 2 years of data. I think today he's going to focus on the 2020 data. We have a little bit more from 2021. So with that, I will turn it over to Todd, and hopefully he can maybe first check that his screen share will work, and then go from there.
Thanks, Dr. Hale. Can everybody hear me? Okay, good deal. Let's see, screen share. All right. So can everybody see that okay? Okay, great. Okay, so yeah, everybody, yeah, my name is Todd Brasseur, and yeah, today I'm just going to talk about the research I've been doing with pitcher plants the last 2 years.
Todd, this is Jen. Can you put yourself in presenter mode? I think it would be easier for us to see.
Okay.
Under slideshow.
I'll
Play, yeah.
presenter viewer. Yeah.
Sorry, from start.
A play from start. Okay.
That's great.
Okay, excellent. Okay, so right now I'm just gonna talk a little bit about hybridizations and like the dangers of hybridization and the benefits of hybridization. So So right now, so hybridization is common in plants, but it raises issues that conservation managers must address when this phenomenon happens in rare or threatened species. So often when species of plants hybridize, they form genetic and sometimes phenotypic intermediates. And this can cause confusion when managers are trying to identify species. Further, distinguishing hybrids from parental species could become more difficult when species hybridize over multiple generations. So early generations often have morphological features that are intermediate between the two parental species, whereas later generations can show features more similar to parental forms. So therefore, conservationists need to recognize varying phenotypes over multiple generations. Hybridization is of special concern for rare and threatened plant species because there could be detrimental effects when this phenomenon occurs. So this is a picture of the first artificial hybridized plant that's created by Thomas Fairchild, which is it's a cross between a carnation and the sweet William plant and it is called the Fairchild's mule. There are many examples of how the dangers of hybridization can affect the fitness of a plant species. Loss of genetic identity through integration is a common occurrence. Integration can leave native species and their habitats vulnerable to invasion by non-natives. So when invasive plants utilize the resources that native plants need, this creates a downfall of the fitness of the native plants and makes it harder for them to reestablish a healthy population. Another example of the dangers of hybridization is if alleles are introduced from outside of the rare plants community, and this could lead to loss of local adaptation for rare and threatened species. So hybridization has also become a problem if native species hybridize with aggressive taxa and their hybrids reduce the growth or replace the native species. If the native species is outsourced by the hybridized individuals, then this will allow the exotic taxa to spread. So although the dangers of hybridization are evident, there are ways in which hybridization is beneficial. An example of how hybridization can aid a rare plant's population is by introducing new genetic variation via gene flow. Gene flow between rare plants and more abundant sympatric species can help maintain the abundance of rare species because it introduces alleles that may facilitate adaptation to changing conditions, keeping the species population thriving. So one example of hybridization benefiting rare plant species was done using the Ranunculus raptans, which is pictured on the right here. And in this study, the scientists focused on the role of long-term population size, genetic distance between populations, and ecological conditions to monitor the performance of 2 generations of offspring. They found that interpopulation outbreeding positively affected the measure of fitness, and fitness superiority was maintained in the second offspring generation. The small populations benefited more from interpopulation outbreeding. And overall, the researchers saw that the benefits of interpopulation outbreeding outweighed the potential drawbacks that happen to populations that experience inbreeding. So when conservation managers are studying hybrids in the field, there can be confusion that arises. So recognizing hybrids morphologically, one would assume that the individuals will represent intermediate characteristics of their parents. This is not always the case because hybrids can show a myriad of their parents' phenotype. So conservationists could identify hybrids using genetic markers such as PCR applications, but these are more useful in ex situ situations and would not be applicable in the field. In order for managers to easily identify hybrids and their parents in the field, a composite metric needs to be administered. This composite metric will assist researchers in the field to easily identify certain rare taxa of plants by measuring physical characteristics that can easily identify the hybrids. So the species, the taxa that were focused on in this study were the sweet mountain pitcher plant, Serosonia jonesii, and the mountain purple pitcher plant, which is a Serosonia Puerpria, Bremer's, Montana. Serosonia Puerpria and Serosonia jonesii are native to the bogs of southern Appalachian mountains. The two species of Serosonia are of concern to conservationists because of the fragile environment these plants inhabit is becoming limited, which is depleting their populations. Serosonia jonesii is federally listed as endangered, and its range is restricted to North Carolina and South Carolina. And Serosonia Puerpria is a federal species of concern, where its range is more spread out. However, the mountain Puerpria varieties range is limited to Western North Carolina. Serosonia jonesii is being actively managed through daylighting efforts, captive breeding, and reintroductions. These two species are known to hybridize, which will have important implications for reintroduction and management. So therefore, it is crucial for conservation managers to recognize the hybrids from the parents and be able to monitor localities for, the hybridization. Okay. So the goal of this experiment is the morphology of these carnivorous plants is an important characteristic study in order to recognize hybridization between the two species and developments of a metric to help conservationists easily identify the two species of Serosonia and their hybrids in the field, and that was the main goal. And so we conducted research at two sites, and we categorized the plants as hybrids, Serosonia Puerpria and Serosonia jonesii, and we measured 8 morphological characteristics. And I can show you those. So you can see the morphological characteristics right here. So we measured the rib max, the aperture perpendicular, aperture width, the hood height, as well as the posterior height, front height, and hood angle. And so for what we did was we used a principal components analysis, and from this analysis, we selected the measures that had the most distinctive morphological features. And from the Pca analysis, we identified the top 3 morphological features, morphological measured features, and used these measures to develop and test 3 simplified metrics. And you can see over here, this shows that the plant's morphological traits form 2 major clusters that differed significantly in PC1. One cluster was distinguished by tall, narrow pitchers with hoods angled tightly over the pitcher apertures, whereas the other cluster was distinguished by shorter, broader pitchers with hoods angled away from the pitcher aperture, and the hybrids being intermediate. These clusters mostly, but not entirely, corresponded to our a priori identification of individual Serosonia Puerpria and Serosonia jonesii plants. And you can see in this figure here that our main objective was to find clumping in the 3 plant groups, and we can clearly see that there is clumping for Serosonia Puerpria and Serosonia jonesii. In addition, the 2 hybrids we identified fell intermediate. So this shows the, let's see, I'm sorry. Okay, so here is the three metrics that we studied, and it shows that although it clearly shows that, sorry, hold on a second. Okay, so these results, they say that it clearly shows that the three plant groups are morphologically different, and the PCA plot analysis is not practical in the field, which is why we wanted to focus on simple metrics to distinguish him. And to do that, we looked at the loadings to identify morphological measures that had the most variation. So we used picture front height, picture post height, hood angle, hood width, because they had the largest magnitude. Since picture front height and picture post height is pretty much the same measurements, we just decided to use front height for two of our three metrics. And the third metric you see, I'm sorry, the first metric you see on the left says the first metric we tried was the ratio between picture front height and hood angle, and hood width. And it shows that there was no significant difference in Serosonia purpurea from the three plant groups. And the front height and the hood angle, this metric showed that there was no significant difference between Serosonia jonesii from the three plant groups. And then the last metric you see on the right there was the ratio between picture front height and hood width. And that showed a significant difference between all three of the plant groups. So we believe that would be the best metric to use in the field. So the principal components analysis test verified that the three plant groups showed morphological differences. This test focused on distinct morphological features and placed each plant into different clusters in the principal components. The posterior height of the picture in Serosonia jonesii was the most distinct morphological trait for that species. The hood width, rib width, and hood angle were the most distinct morphological traits for Serosonia purpurea. And the morphological traits that showed the most intermediate features were the aperture width, aperture perpendicular, and the hood height, which all three plant groups displayed similarly. The phenotype features that distinguish the three plant groups significantly were placed in principal component 1, and the physical characteristics that did not distinguish the three taxa were placed in principal components number 2. So a PCA analysis, it is a very effective tool to accurately measure the morphological features of the three taxa, but it's not really pragmatic for making quick identification in the field. In order for measurements to be easier for managers to apply, we developed and validated three metrics that each focused on two morphological traits. And out of the three metrics, we found that the last metric, which is the ratio between picture front height and hood width, showed all three taxa differed the most, thus distinguishing between the different species and the hybrids. So yeah, then the next step, which we just got from Claudia's work, was we did find genetic identities for these three plant taxa. But I guess like the next step would also be to try to conserve like these bogs that these plants thrive in right now, just so we can further our studies with their morphological features and conserving their genetics and everything. So so that's it. Yeah, I want to thank Dr. Hale and Dr. Ward and everybody that just helped me out through this research that I've been doing. I've loved every minute of it, and I'm extremely proud of what I've done. So.
Great, thank you.
You're welcome.
We'll open it up to some questions. We have one in the chat again. Curious to know the sample size for this study.
I don't have that off the top of my head. I think it was for the one of the sites, the Puerpria was probably, I would say, 30 to 32 different specimens, and the Jonesii was around 7 or 8, and then the hybrids were probably 2. And then for McClure's bog, it was 35 or 40 Jonesii and 10 to 12 Puerpria, and like probably again 2 or 3 hybrids that we found.
Great, thank you. I had a kind of a 2-part question, was just based off of your discussion section that you were just going through, was what are the morphological differences between the pure species and the hybrid species? And then with those differences, what is the scale of that measurement of difference that you were talking about with your box graphs? You know, are we talking like a few millimeters, something that would be easy to kind of see, or is it, you know, really, really tiny differences?
So it's the differences are actually, we measure them in centimeters and then in millimeters, so they're pretty easy to spot out. The hybrids, for the most part, they were like the rib width would be a lot thicker on the Jonesii. It would have like a Puerpria rib width with like a Jonesii plant, and then also, does that make sense? And also the hood height would also be sometimes a little bit smaller on the Puerpria, which would also like which also indicate that would be a hybrid with the Jonesii. And also the pitcher height, we would have really tall pitchers, but the width would be of like a Puerpria. So the Jonesii, the pure species, are very slender, tall pitchers, and then the Puerperias are very kind of they're smaller, more wide. So like I said, it would be the mix between those 2 features.
Great, thank you.
You're welcome.
Does anybody else have any questions? I'll open up the floor. Nobody else has questions. I have plenty of questions. This one's kind of more directed at the whole research group, so it's a more general question. So if whoever wants to answer, multi-part, whatever you want to do. My curiosity is around general conservation goals. So is hybridization less dangerous due to the pollinator species being a generalist? And then what are some unforeseen ecological effects, you know, of hybridization? You talk about, you know, you don't want these species to hybridize, but what happens if they just all hybridize as a natural kind of crossing point? You know, what are some of those ecological effects?
I can answer the second part of that, the ecological, at least some hypotheses about that. So early on in our work with the pitcher plants, we were interested exactly in that ecological question. And one of the really fascinating things that differs between the 2 species is that the purple pitcher plant is a low, broad pitcher, and its hood angle is very high, and it collects rainwater, and it's like a little bowl. Sort of, it's not really that broad, but comparatively, where the other one's really tall and skinny, the hood is over the aperture and water doesn't get in. And one of the consequences of the purple pitcher collecting water is that it fills with this really interesting community of invertebrates and bacteria and other creatures that create this kind of nutrient source for the plant. But also, many of those species are specific to the pitcher plants and are quite rare if they, because they're only associated with pitcher plants. So one of our questions early on was, if you have this species that harbors a community hybridizing with a species that digests everything that falls in it, what is that going to do in terms of creating habitat or maintaining habitat for the species that are obligate pitcher plant commensals? And so what we found in that work was that hybrids tended to have a smaller community, well, maybe not a smaller community, maybe the same set of taxa, but they had a lot fewer organisms living inside them. And we went down an avenue of our research trying to figure out why is that? Is that because mosquitoes, for example, and midges are less likely to lay their eggs in the hybrids? Or is it because maybe they lay eggs at the same, so maybe I should back up. There's a mosquito that's a specialist at pitcher plants. There's a midge, which is a fly, another fly, also a specialist. Lots of bacteria. I don't know to what extent they're specialists. There are mites that colonize. And so it could be that, and so all of those, well, certainly the flies, the mosquito and the midges, the adults are laying their eggs in the pitchers. That's how they're getting there. And so maybe they're laying their eggs at the same rate, but they're not surviving because there's some biochemistry that's different in a hybrid than it is in a Puerpria. And we never really figured that out. We had a student early on do kind of a clever study where he swapped the water that was in hybrids versus in pure, well, what we thought was pure Puerpria, to see if maybe biochemically the colonists could tell a difference when they were laying eggs. And he found that even when you switched the water, you got more organisms in the pitchers that were purple pitcher plant pitchers more than in the hybrids. So that was really interesting. It suggests there might be visual or maybe volatiles coming off of the pitcher itself that colonists are paying attention to. So that's one potential risk that actually personally was why I was really interested in this system to begin with. What does it mean if they're hybridizing? Is that going to mean a loss of habitat for these specialist invertebrates? And I might turn that over to Jen for the part about pollinators, because she's much more a specialist on that front.
Thank you for your answer. That was great.
We also know, so we know that North American pitcher plants do form hybrid swarms, which is like the two species are there and a whole bunch of first generation and later generation hybrids. And we know that that happens when the two species co-occur. It doesn't seem that the two species that are our focus usually co-occur. So they mostly seem to have been put in sympatry, put together by people. And so that's where it gets a little tricky, because there don't seem to be many barriers to cross-pollination. And folks who do pitcher plants for hobbyists, they have observed this for a while. So we do know that, for instance, the hybrid plants make a lot of pollen. We know that they make fewer ovules and fewer seeds than the two parent species, but there seems to be potential for making these hybrid swarms. And really, it comes down to practical stuff like, what do we do with these hybrids? Can we conserve the plants? And like Claudia was talking about, their associated habitat, and Todd said this too, right? Like, can we save the habitat if we don't have a reason to save the plants in them? And if everything turns into a hybrid, under the Endangered Species Act, it ends up being up to one individual to decide what happens to those hybrids, and it just gets a little sticky. So I would say, like, I love hybrids, and when you're talking about hybrids between rare species, it just gets a little a little trickier to try to protect them. So I think we don't know the long-term implications because some of these hybrids have, some of the two parent species have only been put together more recently. And so we don't know the long-term population trajectory within those bogs and what's going to happen. So I think it's great that we have data from Todd and data from Claudia that lets us at least identify so we can census that going forward, right? So we can figure out if the numbers, the relative numbers, vary over time or not.
Quite a complex question to try and tackle. Thank you.
Yeah.
I also, oh, sorry. Also want to throw out there, give Todd some creds for, or props, whatever the lingo is, that he gave that presentation off of a draft version because he had technical issues finding a more recent one. But I found a shortened version of his more recent talk. Sorry, Todd, I didn't find that earlier. But if you don't mind, it's got some great pictures of the plants in it. So I'm going to share my screen because it, well, now it's telling me I can't. Let's see.
Stephen, will you make her a co-host real quick? There you go.
Okay,
That'll
thanks.
be good now.
To the chat question, much of this habitat has been lost in Western North Carolina specifically. The mountain bogs, what's it called, Becca? Mountain Bogs Network? Mountain
Oh, the
Bogs?
Bog Learning Network.
No, the like new federal.
Oh, Mountain Bogs Refuge. It's a national wildlife refuge.
Yeah, Mountain Bogs National Wildlife Refuge. If you look that up, they have some statistics about how many bogs have been lost. So I'll see if I can find that and put the link in the chat.
Yeah, so this image, I can't see my Zoom anymore. So if someone's trying to talk, I'll just for a second here. This is the Joneses or the Mountain Sweet pitcher plant. You can tell it's really tall and skinny. The hood angle is a little bit more over the pitcher, whereas this is the purple pitcher plant. And the hood is on many of them, depending on how they're actually situated on the ground, it's just wide open to the air, and they're like a little vase, a little bowl, just collecting water, and you know, mosquitoes and midges come in there and lay their eggs. So this is capturing, as Todd said, the kind of short, squat pictures of a Serosonia Puerpria, and this cloud is capturing the tall, skinny pictures like these.
Great, thank you. That definitely helps my mental picture. All right, open it up one more time if there's any more questions. But if not, big round of applause for our presenters.
Thank you.
Thank you to the UNCA professors who have mentored them.
Thanks for having us all. We really appreciate the opportunity to let our students share.
Yeah, if you got any more students in the pipeline, send them our way. We'd love to have them. Keep the pitcher plant research coming. Sounds like really interesting stuff, especially now that you've gotten a little bit more meat of the research done. So good stuff. Thank you.
Thank you.
Thank you.
Thank you.
Thanks very much.
And stay tuned for next month. We're not sure what we're doing next month, whether we're having a talk or not. So if
We
you know
are.
of
Jen's
anybody,
giving a talk next month.
oh, good. I forgot about that. Excellent. Thank you, Jen. Appreciate it.
We're
right, well,
continuing the pitcher plant saga.
Excellent. Well, we'll see you all next month, hopefully. All right. Thanks.
Thank you.
Good night.